I am Nagashree Seetharamu, thoracic oncologist at Northwell Health in New York. Next slide, please. These are my disclosures. Next slide, please. Okay, so I am going to be talking to you about NTRK, or neurotropic tropomyosin receptor kinase gene fusions and TRK proteins. I just want to walk you through normalcy and cancer. So the TRK genes, or the NTRK genes, are important for neuronal development, for nervous system development. They are expressed in normal nerve tissue, and they have roles in development of nervous system and nervous system function. So on the right-hand side, I have a figure that tells you about the three genes that we'll be focusing on, the NTRK1, NTRK2, and NTRK3. They are on different chromosomes, and they don't have, they're not close to each other. They're on different chromosomes. And they result in manufacture of proteins called TRKA, TRKB, and TRKC. And as I said, they are important normal tissues. However, sometimes these genes can either flip over, jump over to another gene, and cause what we call as chimeric fusion proteins, whereas a part of the NTRK gene links with another gene and creates a fusion protein, a fusion gene that can lead to hyperactivation of the system, and I'll show that in the next slide. So the TRKA, thank you. As I mentioned here, you see in the middle, you have the TRKB for an example. So when you talk about the receptors, these are molecules on the surface of the cell. For activation to happen, for the signals to be transmitted from outside to inside the cell, resulting in cell growth and proliferation and going to other places, you need the two receptors to come together. So it's called dimerization. So the dimerization happens when there is a signal from outside. The signal is called a ligand. So that when that happens, there's triggering of downstream pathways, as you can see here, the RAS pathway, the PI3 kinase pathway, and the PLC gamma. The PLC gamma is important for neuronal differentiation. The other two pathways are important for cell differentiation and proliferation, meaning it's important for cell growth, the cell to divide and go to other places. So when the flip happens, you don't need the ligand anymore. The activation happens without any ligand necessary. Next slide, please. Sorry about that. Okay, so I just want to talk to you about the NTRK fusions and other NTRK gene variants. The fusions are important, but there are also other mutations and other variations that can happen with NTRK. So generally, the fusions are formed, as I said, either by flipping of the NTRK gene, NTRK1, 2, or 3, when the NTRK sequence is the 3', one of the ends of the gene it just jumps into another gene or 5' sequences of other genes. The result is what is called an oncoprotein that causes the TRK kinase ligand independent activation, meaning you don't need the ligand anymore. The pathway is open, it's activated, just the dimerization is constantly there, and that leads to cell transformation, proliferation, and other invasiveness, going to other places and all that stuff. However, it should be noted that all fusion partners they do not have typical dimerization domains, and not all structural rearrangements result in productive fusions. So not all rearrangements result in in pathogenesis or in cancer development. And not all, and definitely not mutations or gene amplifications, they have not validated to be targets for treatment. So it's important to know that when you're reading, when we're reading a report. Next slide, please. Next slide, please. Oh, so this is the timeline of discovery. I mean, back in 1982 was when the first fusion was detected. This was the TPM3 NTRK1 in colon cancer specimen. It took so many decades for it to be recognized as an oncoprotein and for it to be targeted for medication.